Why are intestine epithelial cells so different compared to something like neurons? What differentiates them, and what are some misconceptions about them? There is a common misconception that different cells have different genomes because they are so physically different. This misconception is completely wrong. In fact, virtually every single cell in your body has the same DNA sequence, and in fact uses a principle called gene expression to differentiate. So let's fix this. Every single cell has the same DNA sequence, unless mutated. But, first of all, how do we know this is the case? Since the late 20th century, we've had solid proof for it. Researchers have come up with tons of ways to prove identical genome in all cells of an organism, but the most notorious case was Dolly the sheep, where in 1995, Roslin Institute researchers took a differentiated cell nucleus and all of its genetic information from a developed sheep's udder and implanted the genetic information inside of an egg to create a baby sheep. This sheep was completely healthy and devoid of mutation. If there was different DNA sequence within the egg, the embryo would likely die or experience physical or mental abnormalities, demonstrating that the genetic information in both cells must have been the same. So how do cells develop into wildly different tissues? Well, it's due to a process called gene expression. What is gene expression exactly? Gene expression is the process by which information encoded in a gene is used to synthesize a functional gene product, typically a protein or RNA molecule. Gene expression can be divided into two actions: post-translational modifications, which happen while or after the protein is made, and transcriptional regulation, which happens before or during the RNA transcription. One method of gene expression is post-translational modifications, which include phosphorylation, acetylation, and other methods in order to rapidly modify a protein's function in the cell and stimulate or inhibit its respective activity. The other method of gene expression is transcriptional regulation, which occurs while or before the protein is transcribed and is used for more long-term stimuli. One type of this is histone remodeling complexes. Histone remodeling complexes affect gene expression by using ATP-dependent energy to alter the positioning of nucleosomes, including addition of acetyl groups to histones, which decondenses chromatin and facilitates transcriptional machinery access to gene promoters, leading to increased gene expression. Another method is the use of transcription factors. Transcription factors are proteins that bind to specific DNA sequences near gene promoters, acting as activators or repressors to regulate gene expression. Repressors can inhibit RNA polymerase by binding to gene promoters and blocking the assembly of the transcriptional machinery, and some repressors can be phosphorylated, altering their activity and further inhibiting gene transcription. To summarize, gene expression is the hidden credit for the 400 different cell types in our body, and virtually every cell we have shares the same genome regardless.